Good morning, students. Last class we have discussed about the five kingdom system of classification proposed by R.H. Taylor. All the living beings are divided into two kingdoms by Carlos Linnaeus. These are Plantae and Animalia. And Haeckel proposed three kingdom system classification. These are Protista, Plantae, Animalia. Copeland proposed four kingdom system classification. These are Monera, Protista, Plantae, Animalia. And R.H. Taylor proposed five kingdom system classification. These are Monera, Protista, Fungi, Plantae, and Animalia. Copeland actually named the term Mycota. Copeland proposed four kingdom. These are Monera, Protista, Plantae, Animalia. But Copeland given the term Mycota, and the term Monera was given by Dr. Allen in 1960. Kingdom Monera is a large group. It also divides into eubacteria and archaebacteria. Kingdom Monera includes all unicellular prokaryotes. It includes all unicellular prokaryotes divided into two groups, eubacteria and archaebacteria. Under the eubacteria, a number of groups are found: bacteria, cyanobacteria, mycoplasma, rickettsia, actinomycetes, etc. These are bacteria, cyanobacteria, mycoplasma, rickettsia, and actinomycetes. All these groups are unicellular and prokaryotes. This is the property of the Monera. The kingdom system classification. In the kingdom system classification, all unicellular and prokaryotes includes in the kingdom Monera. Now we will discuss about the bacteria: structure, classification, reproduction, nutrition, and economic uses of bacteria. Bacteria is unicellular prokaryotes. Unicellular, this is the structure of the bacterial cell. Unicellular and prokaryotes. It is surrounded by a rigid cell wall. The cell wall of bacteria is different from that of the plant cell. In case of plant cell, the cell wall is composed of cellulose, but in case of bacteria, the cell wall is composed of peptidoglycan. The cell wall is composed of peptidoglycan. Here, peptidoglycan, not cellulose. In the peptidoglycan, N-acetylmuramic acid, N-acetylglucosamine, N-acetylmuramic acid, and N-acetylglucosamine. These are the amino sugars. The N-acetylmuramic acid and N-acetylglucosamine are connected by glycosidic bond. These are connected by glycosidic bond to form a straight chain. From the N-acetylmuramic acid, the amino acids are formed. They are connected by peptide bond to form the skeleton of the cell wall. Clear? These N-acetylmuramic acid and N-acetylglucosamine, these are the amino sugars connected by glycosidic bond, and two rows of amino sugars are connected by peptide bond to form the skeleton of the cell wall of bacteria. Above the cell wall, in few bacteria, capsule present. The capsule may be thick or may be thin. These are the microcapsule or macrocapsule. The capsule gives protection to the bacteria from the adverse conditions. Below the capsule, cell membrane present. In case of bacteria, the cell membrane is folded in few places called the mesosome. The cell membrane is folded in few places called the mesosome. The mesosome helps in respiration. It helps in DNA replication, as well as it helps in septa formation. So it is the most important part of the bacterial cell. The cell membrane encloses the fluid called the cytoplasm. The genetic material of the bacteria is single circular histone 3 DNA. It is single single circular histone free DNA. That is single circular histone-free DNA. But in case of other cells like eukaryotic cells, the DNA are linear. The DNA are linear with histone protein. Here the DNA is single circular histone-free, always attached to the mesosome, and such type of nucleus is called incipient nucleus or nucleoid or genome. Clear? Such type of nucleus is called incipient nucleus. Here organized nucleus is absent. Organized nucleus means nucleus with nuclear membrane and nucleus are absent. Such type of nucleus is incipient nucleus or nucleoid. Besides nucleus, small extranuclear DNA is present, may be present, called plasmid. Besides this incipient nucleus, some extranuclear DNA, that is called plasmid, may be present. The plasmid may be F plasmid or R plasmid or called plasmid. The plasmid performs some special functions. F plasmid means fertility. The bacteria having F plasmid behaves as male or donor, and without plasmid behaves as female or recipient. Similarly, R plasmid resistance, and that gives resistance against different types of antibiotics, and called plasmid produces cholecinogen having some antibiotic properties. Now we study the structural parts of the bacterial cell. It is unicellular, prokaryotic, having cell wall surrounded by capsule. Below the cell wall, cell membrane present, which encloses cytoplasm. The cytoplasm contains 70S ribosome. The ribosomes are two types, 70S and 80S. In eukaryotic cell, both 70 and 80S ribosomes are found, but whereas in prokaryotic cell, only 70S ribosome is found. The 70S ribosomes, it is the site of protein synthesis. Incipient nucleus present, plasmid may be present, and RNAs are present, result food present. Generally, the result food is the glycogen. On the surface, a number of small appendages are found, called the pili and fimbriae. Pili and fimbriae are present. The pili, composed of pilin protein, helps in sexual reproduction, helps in formation of conjugation tube during the sexual reproduction, whereas the fimbriae, it is also a smaller structure, helps in attachment on the object. The larger extensions are called flagella. In case of eukaryotic cell, the flagella shows 9+2 arrangement. These are special arrangement we will discuss on later classes, the 9+2 arrangement. But in case of prokaryotic cell in bacteria, the flagella does not show the 9+2 arrangement of microtubules. The flagella helps in locomotion. Now we will discuss about different types of Monera or bacterial cell. According to the shape and size, the bacterial cells are different types. Number 1, cocos. Cocos means spherical. Maybe 1 cell present, maybe 2 cells present, maybe 4 cells present, maybe 8 or 64 cells present in a cubical manner. Maybe 8 or 64 cells, 3-dimensional structure in a cubical manner. Maybe a chain of cells are present, or a group of cells are present, Cocos means circular. If a single cell present, called the monococcus or micrococcus. 2 cells are present, called diplococcus. 4 cells are present, called tetra. 8 or 64 cells are present in a 3-dimensional structure called sarcina. A chain of cells are present called streptococcus. Streptococcus, and a group of cells are present called the staphylococcus. Clear? Group of cells are present called the staphylococcus. Next, bacillus. Bacilli, it is the rod-shaped bacteria. If a single cell present called bacillus, two cells present called diplobacillus, a number of cells are arranged one above the other called the palisade bacillus, and if a chain of cells are present called the streptobacillus. Generally, the bacillus are rod-shaped with blunt end. Single cell, bacillus, two cells, diplobacillus, a group of cells arranged one on the other, palisade bacillus, and a chain of cells called the streptobacillus. If flagella found on the bacillus called trichos. If any flagella is absent, called atrichos. If single flagella present on one end, called monotrichos. If single flagella present on both ends, called amphitrichos. If a group of flagella present on one end, called cephalotrichos, and group of flagella present on both the ends, called lophotrichos. If flagella found throughout the surface, called peritrichos. Clear? Based on the presence of flagella and the number of flagella, it's different types. If flagella is absent, called atrichos. If one flagella present on the one end, called monotrichos. Two flagella, one on each end, called amphitrichos. Group of flagella on one end, called cephalotrichos. Group of flagella on both ends, called lophotrichos. And flagella throughout the surface, called the peritrichos or polytrichos. Now, next type of is the spirillum. Generally, it is coiled like a screw. Called spirillum. And last one is the comma. Example is the vibrocomma. It is comma set. Clear? Now structurally it is unicellular, prokaryotes. According to the cell, it may be coccus, bacilli, spirillum, comma, and according to the presence of flagella, it is of different types. Now we will study about the types of nutrition of bacteria. We will study the types of nutrition of bacteria. Clear? According to the nutrition, the bacteria are divided into two groups. According to the nutrition, bacteria are divided into two groups: autotrophic and heterotrophic. Clear? Autotrophic and heterotrophic. Autotrophic means the bacteria can synthesize their food materials. The bacteria which can synthesize their food material called autotrophic bacteria. Autotrophic again divides into photoautotrophic and chemoautotrophic. Clear? Autotrophic means the bacteria which can synthesize all their own food material. Autotrophic, if it synthesize their food material in the presence of sunlight, called phototrophic, or from chemicals called chemoautotrophic. The phototrophic may be green sulfur bacteria or purple sulfur bacteria. In prokaryotic cell, any membrane bound organelle is absent. In prokaryotic cell, any membrane bound organelle like mitochondria, chloroplast, Golgi body, endothelial reticulum, etc. all are absent. Clear? Only ribosome present, which is 7 S type. If the mitochondria is absent here, the function of mitochondria is performed by mesosome and chloroplast is absent. The chloroplast is a double membrane organelle, here it is absent. But beside the chloroplast, here some chromatophores present. Here in few bacteria, some chromatophores present. That chromatophore pigment helps in photosynthesis. But this type of photosynthesis is anoxygenic photosynthesis. Means in case of plant, the in case of plant, carbon dioxide combines to water to form glucose, and here oxygen is removed out. Oxygen is a byproduct, is released out. But in case of bacteria or in case of prokaryotic cell, it is anoxygenic photosynthesis. That is, instead of water, here hydrogen sulfide or sulfur containing compound is used. Sulfur containing compound is used. So your oxygen is not released out. That is called anoxygenic photosynthesis. Autotrophic bacteria, two types: phototrophic, chemotrophic. In the phototrophic, bacterial chlorophyll present and pigment present. That is called bacterial chlorophyll. These are two types: green sulfur bacteria, purple sulfur bacteria. They perform the photosynthesis. That is anoxygenic photosynthesis. Here oxygen is not released out. Here hydrogen donor is the hydrogen sulfide or any sulfur containing compound, not water. In case of higher plants, water is used and oxygen is evolved. But in bacteria, sulfur containing compound is used and sulfur is released as a byproduct, not oxygen. So this type of photosynthesis is called anoxygenic photosynthesis. If the bacteria extract the energy from chemicals called autotrophic bacteria, clear? Chemotrophic bacteria. It is the two types that depends upon the types of the chemicals used. If it extracts its energy from inorganic compound called chemolithotroph or organic compound called chemoorganotroph. Clear? The chemoautotroph bacteria are two types: chemolithotroph. Here the bacteria extract energy from the inorganic compounds called chemolithotroph, and chemoorganotroph, it uses organic compound for extraction of energy. Here the nutrition two types: autotrophic and heterotrophic. Autotrophic and two types: phototrophic and chemotrophic. Here pigments are present, that is called bacterial chlorophyll, and by the presence of sunlight can prepare the food material, but that is anoxygenic photosynthesis. Here it extracts the nutrients, energy from the chemicals. Again, heterotrophic bacteria is of two types. Photoheterotrophic and chemoheterotrophic. Photoheterotrophic, this bacteria possesses bacterial chlorophyll and can trap the solar radiation, but for carbon source depends upon others, so it is mode of heterotrophic nutrition. Depends upon others for carbon source, so it is heterotrophic mode of nutrition called photoheterotrophic. The chemoheterotrophic again three types. These are saprophyte. Second is the parasite, and third is the symbiotic. It is of three types: saprophytic, parasitic, and symbiotic. Saprophytic, parasitic, and symbiotic. Saprophytic means they depend upon other dead decomposed materials for the nutrients. They depend upon other dead bodies for the nutrients. Parasite, they depend upon other living beings for the nutrients. That is host. And symbiotic, these are the mutual relationship between two organisms. The saprophytes again are two types: obligate saprophyte and facultative saprophyte. It is also two types: obligate saprophyte and facultative saprophyte. Means obligate saprophyte means these are only saprophyte. Obligate saprophyte is a saprophyte which lives only on the decomposed material. But the facultative saprophyte is not a saprophyte, it is a parasite. facultative saprophyte is not a saprophyte, it is a parasite. In the absence of the host, it may depends upon it lives saprophytically. It is the facultative saprophyte actually is a parasite, but in the absence of the host, it develops saprophytically. Similarly, parasite also two types: obligate parasite and facultative parasite. The obligate parasite is the only parasite. Obligate parasite is only parasite, whereas the facultative parasite is not a parasite, it is a saprophyte. The facultative parasite is not a parasite, it is a saprophyte, and in the absence of the decomposed materials, it may live parasitically. Last one is the symbiote. Symbiotic relationship means mutualism. Two organisms live together and both are beneficial to each other. Two organisms live together and both are beneficial to each other. Best example is the Escherichia coli. Best example is the E. coli in human intestine and rhizobium bacteria in leguminous plant. Rhizobium bacteria, root of leguminous plant, and Escherichia coli in human intestine. This relationship is called symbiotic. In our elementary canal colon, bacteria found called Escherichia coli. This Escherichia coli provides us vitamin K, vitamin B12. In turn, we provide nutrients to the bacteria and give shelter to the bacteria. Similarly, the rhizobium bacteria fixes the atmospheric nitrogen inside the root of leguminous plant. This relationship is called symbiotic relationship. So, according to the nutrition, the bacteria are two types: autotrophic and heterotrophic. Autotrophic can prepare the food material. If we prepare the food material in the presence of sunlight, called the phototrophic, or from chemicals, called chemotrophic. The chemotrophic, if the chemical is inorganic compound, called chemolithotroph, and organic compound called chemoorganotroph. Similarly, heterotroph two types: photoheterotroph and chemoheterotroph. Clear? In the photoheterotroph, actually, this bacteria possess chlorophyll and can trap the solar radiation. But for carbon source, it depends upon other organisms. So it is the mode of nutrition is heterotrophic nutrition. The chemoheterotroph is of three types: saprophyte, parasite, and symbiotic. The saprophyte is of two types: obligate saprophyte means only saprophyte. Facultative saprophyte, it is not a saprophyte, it is a parasite, but in the absence of the host can develop saprophytically. Similarly, parasite is of two types: obligate and facultative. Parasite means that depends upon the living mix of the host for their survival. If the bacteria depends only on the host called the obligate parasite. If the bacteria depends upon the dead bodies, in the absence of dead body, if it lives parasitically called facultative parasite. Symbiotism or mutualism, it is of two types. Several examples are found in the symbiotic relationship. Here, two organisms live symbiotically, both are beneficial to each other. The best example is the Escherichia coli and human intestine. This bacteria live in the colon of the man and synthesizes the vitamin K and some B12. Second example is the rhizobium bacteria, which live inside the root of leguminous plant and fix the atmospheric nitrogen. Clear? This is all about the structure, classification and nutrition of Monera. Next class we will discuss about the reproduction, asexual reproduction, sexual reproduction and economic uses of bacteria. Thank you.